Scientists have known that dead cells cause Alzheimer's for a while now, but the origin was unknown until recently. When a human cell gets old or damaged, it usually destroys itself safely using a common process called apoptosis. However, in a disease like Alzheimer's and FTD, traditional apoptosis wasn't happening enough to explain the massive decay and widespread loss of brain cells that causes the memory loss. When toxic waste clumps up together inside of a neuron, it creates a state of cellular panic known as proteotic stress. This stress activates an enzyme called p38 MAP kinase. Instead of the cell cleaning up, this enzyme malfunctions and directly attacks that lamin B1 protein. This critical structure or protein holds the cell's nucleus together. Without its protective structural wall, the nucleus literally warps up and shrivels. Because the researchers successfully mapped out the exact chemical pathway of karyoptosis, they were able to experiment with blocking specific interactions between the p38 enzyme and the lamin B1 protein. When they introduced the target compound to block the switch in laboratory models, nuclear disintegration was successfully halted, saving neurons from decaying. 35% of neurons in the frontal cortex of Alzheimer patients showed active, shriveled markers of karyoptosis. In contrast, only 15% in healthy, aged brains showed similar signs. While it's still early to call this a cure to Alzheimer's and other related diseases, it could very well be a possible contribution to its solution.